Now it's 2 o'clock. Good afternoon, audience. Welcome back to the symposium. This is James Fang, the chair of the afternoon first section. The first invited speaker in this section is Professor Rudolph Wu, who is a chair professor currently in the Department of Science and Environmental Studies at the Education University of Hong Kong. Professor Wu is a world-leading expert in marine ecotoxicology, in particular in the research area of aquatic hypoxia. Many of us, including me, have been students of Professor Wu. Today, we are fortunate to have Professor Wu here to share his insight into hypoxia in aquatic environment. Professor Wu, please.
Thank you. Thank you for the introduction, James. Now, in this talk, I'll try to discuss the topic about hypoxia. And then if you look at the topic, you'll be sort of maybe surprised that nothing could be worse. I hope within this 30 minutes, I'll be able to convince you that hypoxia is the most important environmental stressor in our environment today. Now, first of all, what is hypoxia? Hypoxia is defined as the dissolved oxygen of the water, which is less than 2.8 milligram oxygen per liter, which is equivalent to 2 milligrams oxygen per liter. Now, we shouldn't blame everything to pollution. Hypoxia may be a natural phenomenon or due to pollution. For example, stratification due to this density difference, like with a thermocline or a halocline, that means a different temperature or different salinity of the water can block the oxygen diffusion, and that causes the hypoxia. And probably more importantly, in recent years, human activities add a lot of nutrients to the water. We all know that the limiting factor of this phytoplankton growth is nutrient. So we put excessive nutrient into the water that causes a phenomena which we all know about. This is eutrophication. And also because we discharge a lot of organic matter in the water, and then the bacteria, when they degrade these organic matters, require oxygen. That would consume a lot of oxygen in the water, making the dissolved oxygen low and become hypoxia. Now, in actual fact, in the last 30 to 80 years, decrease in hypoxia occurred all over the world, in USA, China, Scandinavia, United Kingdom, Sweden, Germany, Denmark, Black Sea, and the Adriatic Seas, all over the world, in actual fact. Now, nowadays, globally, there are more than 400 is what they call dead zones, which is associated with eutrophication. That is to say, these 400 dead zones are due to human activities and causing this eutrophication. And the total area up together is more than 245,000 kilometers square. For those who are not familiar with what the total area means, the total area is much larger than the United Kingdom up together. And then one of the dead zones in the Gulf of Mexico, for example, which occur every summer. And then the size of this dead zone is similar to the whole area of the whole state of Virginia. So this gives you some indication of how bad and how large it is. Now, what's more is that if you look at the number of dead zones, and you see a dramatic increase from something like 1960s to 2000. And then in actual fact, these data is a bit outdated, and then that number of dead zone and the total area of dead zone keep on increasing ever since. Now, because of hypoxia, a lot of major change in structure and function of marine ecosystem over very large area. What I like to emphasize is over a very large area has occurred all over the world, something like in China, in Europe, in Japan, in USA, and similarly some in Australia. Now, there's well-documented data showing that hypoxia actually caused a lot of animals to die, what they call mass mortality. So it's not uncommon to see lots of fish are killed by this low oxygen. Very simple, not enough oxygen, and they all die. And this shows you some picture that's very common. And let alone that they also have, for this natural population of fish, for those who culture fish, again, they suffer a lot because hypoxia can kill off all their sort of culture fish within several hours. And then, of course, this hypoxia also causes death in the natural animals, like coral, and then like a lot of these animals sitting on the ground of these the seashore. Now, in terms of function and structure of the ecosystem, and hypoxia has caused major change in species composition. They actually change the species composition, and later on I'll talk about this and in greater detail. And then also they then decrease in the biodiversity and species richness, and change in the trophic structure and the food web. So that's a very fundamental changes in, as I said before in yesterday's lecture, the ecosystem function is utmost important, and actually is much more important than the structure itself. So it changes the trophic structure, change the species, change the whole function, and change the life cycle and species distribution. And what's even more long-term is that they actually alter the bioirrigation. And some of you may not know that a lot of the animals, like the polychaete worm and then bivalve and things like that, and they're living on the sea bottom, and then they keep sort of stirring the sediments, stirring up the mud, and then but this sort of activity, what they call the bioirrigation, can turn some of these microbial animals and microbial organisms, and then into active form, and then they are sort of responsible for a lot of this essential biogeochemical process and sediment water exchange. So if you kill off this animal or change the animal composition, and then you expect these sort of major biogeochemical cycle will be disturbed. Again, I'll show you some scientific data that we collected later on. Now, long time ago, we have been interested in this and the hypoxia phenomena because we regard this as one of the most important, if not the most important environmental problem globally. So compiling literature and some of the data, we find that during hypoxia, for example, and then you got lots of animals which are suspended feeders. But when changed to hypoxia, basically a lot of these animals change into deposit feeder because suspended feeder need more oxygen. Deposit feeder because they need more deposit and they survive better. So again, there's a major functional change in the functional group of these animals. And then they change from demersal fish under normoxic condition. Demersal fish means fish living on the bottom. And then change to pelagic fish when the water turned into hypoxic condition. Again, that's a fundamental change, mainly because this demersal fish which is living on the bottom, well, because the bottom oxygen depletion would be more serious than that in the water body. So they change from demersal fish to pelagic fish, and from high diversity to low diversity, and then from large body size to smaller body size, mainly because smaller body size can be more able or more efficient to take up oxygen from the water. So again, what these hypothetical changes and is summarizing, there's again a major change in species group and also affecting the function of ecosystems. Now, more recently, funded by the state key level of marine pollution, we conduct some study, and for the first time, we demonstrate that actually hypoxia can actually decrease the sediment carbon fixation. As shown in this figure, what we do is that we transplant the sediment from the hypoxic site to the normoxic site, and from normoxic site to the hypoxic site, and so on. And you see that after several weeks here, and then you transplant the sediment and has decreased in the carbon fixation. In contrast, the hypoxia actually increased the sediment phosphate fixation after 10 weeks. So you see that this, again, is very fundamental. We all know that the carbon, nitrogen, and phosphorus cycle, a most important biogeochemical cycle in the marine environment, not only in the sediment, because in the sediment fixation will affect the water nutrient concentration as well. Now, these shows for the first time that hypoxia can actually affect the geochemical cycle by experimental evidence. Again, some 15 to 20 years earlier, when we start interested in the problem with hypoxia, we actually discover for the first time that hypoxia is an endocrine disruptor. And the potency of endocrine disruption is comparable to the most toxic EDC in the world. Now, what we did is we put the fish under hypoxia condition for several weeks, and then we find that testosterone, which is male hormone, decreased dramatically, while the female hormone, estradiol, increased dramatically. And then, so we said, oh, is it only the carp? So we tried this out in different species, trying silverfish, the same happened. And then also we find out that this hypoxia actually affects this spermatogenesis. That is to say, the development of the male gonad. And you can see that under hypoxia, both the silverfish and common carp, and they all share the commonality is that they actually delay in their sort of the spermatogenesis. You got less spermatic, which is the mature sperm. And then you got more sort of primordial sort of germ cells like the spermatogonia and spermatogonia. And then for the diameter of these lobule, and then under hypoxia, the common carp actually sort of decrease in the diameter of this male lobule, showing that the male reduction and spermatogenesis process has been affected. Then we look at the sperm motility. Sperm motility is a very good indicator on the sperm quality. And then, so you show that the swimming activity, different velocity of the sperm of the fish also decrease. And then we use other models like the zebrafish, and also find that the ovarian cell stage, similar to the spermatogenesis, has been sort of inhibited. So you got less sort of the mature oogonium, the oocytes, as compared with the sort of the premature one under hypoxia, showing that hypoxia actually inhibits the development of this ovarian. Now, at that juncture, a guy came from the USA, said, oh, come on, and this is all laboratory experiment. How do you ensure that it also happens in nature? So he said, oh, I don't know, because as you may probably aware, doing this field validation is very expensive and very tedious, but we can't afford to do that. And then Peter Thomas in Texas actually did similar experiment in the field of Gulf of Mexico, collecting fish, Atlantic croaker, from different places with different hypoxic level. And then it shows exactly the same thing in Atlantic croaker, and showing that in Atlantic croaker is the same. And then the sort of development of oogenesis are inhibited under hypoxia, under field condition. Then, if you put things together, we'll find that under hypoxia, the fertilization rate decreases, mainly because you got decrease in spermatogenesis and you got decreases in oogenesis. So egg is not very good, the sperm is not very good. This is why you decrease in your percentage fertilization from almost 100% to only 55%. Then with this sort of substandard fertilized egg, the hatching rate also dramatically reduced from something like 100% to less than 20%. So even the larvae has been able to hatch, the 14-day larval survival again decreased dramatically by 1/2. Now, if you take everything together, something like compare the sort of normal egg developed to larvae under normoxic condition with the egg produced by the fish affected by hypoxia, from egg to larvae, it decreased from something more than 90% to 5%. So that is this shows that hypoxia has a very dramatic and serious effect on reproduction. something like only 5% of the original sort of egg to larvae ratio can be obtained. Now, the second thing that we demonstrate is that hypoxia is teratogen. Teratogen is a chemical that can make something lead to malformation, abnormal development. So initially, we find that they actually delay the development and then delay the larval development. So that's the larvae after 24 hours under controlled condition. And then after 48 hours, they got tail and things like that. But if you put the zebrafish embryo under hypoxia, well, for the same period of time, 24 hours, you can see that the development has been delayed. There's no tail yet. And then after 48 hours, this guy can almost swim and is still like embryo. Now, then we look at the malformation, primary skeletal deformity and abnormal development. And then you can see that it manifests after 96 hours. That's the control. That's the treatment. That is to say, under normoxia, normal condition, the malformation is less than 10%. Whereas, I mean, not after 98 hours, the malformation goes up to 15. After 160 hours, and then the normal malformation rate is only 10%, while this increased to almost 20%. Again, we use the fluorescent dye to look at the apoptosis. Apoptosis means means when the cell is trying to, to kill off itself, because the cell during development, they can ask that normal cell to sort of go away. Now, that's what we call the apoptosis. Now, hypoxia actually enhance apoptosis in the head, but not the tail, as shown by these fluorescent dyes dyeing. And apoptosis is identified by this staining, and from the head, number of apoptotic cells increase, whereas mean the apoptotic cells in the tail decrease. We all know that apoptosis is an important process in determining the normal development of any animals. So this shows that they actually disturb this process and then therefore leading to this and abnormal development. Now, then again, we talked to a number of scientists over the world. So these guys raised a lot of questions. They said, oh, come on, of course, under hypoxia, a lot of these animals, regardless, will reduce their biological activity, like under not condition, which is suboptimal, they decrease in the activity. So this is why they decrease in the hormone secretion, decrease in the growth, and then sometimes decrease in the development. So maybe it's just as simple as that. Now, and the other guy said, well, the reason why you find a smaller gonad in the hypoxic fish and also find the reduced hormone production, lower hormone production or disturbed hormone production is due to reduce energy intake and reduce growth, because under hypoxic condition, the fish will feed less. So in that case, they got less energy, and this is why they got a smaller gonad, and nothing to do with disturbance. Okay, and then the other, so endocrinology said hypoxia affects sterogenesis. Now you provide us the end result showing that spermatogenesis is disturbed. You also provide data on hormone is disturbed, but how do you know that sterogenesis is disturbed? And then the other guy asked, does hypoxia also affect the GnRH and gonadotropin, not only the sex hormone? Now, it will be very difficult for us to sort of test this condition, mainly because if you expose the fish to hypoxia, well, everything will be affected. So we use another approach. We use an in vitro study using a human adrenocortical carcinoma cell line, and then expose this cell line to hypoxia. We actually show that 3 genes regulating sterogenesis were affected by hypoxia, but more importantly, not the others. Now, this is the sort of the steroidogenesis pathway from cholesterol all the way to testosterone to estradiol, the male hormone and female hormone. And these are the enzymes that regulates all this process. We actually find that they affect specific process rather than specific enzyme like this. They affect the CYP11A, 3-beta-HSD, and then also CYP11B1, and then CYP19. In particular, we demonstrated that if they affect CYP19, so this is why testosterone cannot be transformed in the estradiol, and this is why you got a higher level of testosterone than estradiol. So these demonstrate specifically that under in vitro condition, which is only the gonad is affected by these hypoxia, and then they affect specific process, but not the others like CYP21 and CYP20B1 and so on and so forth. Now, then we look at the problem further. We find that hypoxia actually affects sex differentiation and alters sex determination. So again, we use zebrafish as a model. So after growing the zebrafish for 2 generations under hypoxic and normoxic condition, under normoxic condition, well, you got about 62% male, and then, but under hypoxia, you got almost 75% male. Then, again, we look at the hormone again, look at the hormone level in the female fish and the male fish. We actually compare the testosterone to estradiol level. Now, you find that basically the testosterone to estradiol, that means to say the male to female hormone ratio remain unchanged in the male, but changed in or increased in the female. That is to say, they actually affect the female by increasing the testosterone level as compared with the estradiol level. So the female fish got more male hormone than female hormone, and this is why they direct their sex differentiation into male. Now, we all know that in this world, women and female are much more important than male. And then because they are actually the key factor controlling reproduction and hence the future population. So no girlfriend, big problem. Now, then you can see that if you in the population, you got more and more male and less female, eventually you can predict that the population will extinct, right? Because you got not enough female. No matter how many female you got, does not matter. Now, then we ask the question further. Is the observed male bias phenotypic or genotypic? Because we find that they actually affect the hormone only. So is it phenotypic or genotypic? Then I talked to a medical doctor when we are sort of showcasing our research in the UGC's format. And then they said, oh, we said, oh, maybe it also occurs in human beings. Nonsense. Human being in the sex, human being is determined by the genotype, which is got XX chromosome, XY chromosome. So it's not controlled by the hormone, okay? Or even controlled by hormone is to much less extent. So I asked the question, will the same happen to other species with sex determining chromosome? So the medical doctor said, oh, no, come on, unlikely. And then, so I tried to get one of my previous students to work on the problem. So we can't work on human being, but so we can only select a fish with a sex determination chromosome, just like human being, okay, which is the which we treat that which is we treat this fish and the with under hypoxia and normoxic condition, and then we look at the sex reversal, and we find that even for the genotype XX, the sex reversal is 77%, and for the genotypic XX, is 54%, whereas the genotypic XY to remain unchanged. So this shows very clearly saying that even if you have a sex chromosome of XX, the sex reversal can still occur. Now, then we go back to the medical doctor, and then I also did some more study because, as I said, we couldn't work on human being with no capacity of making human being. So we searched the literature. We find out that patients suffering from sleeping apnea. Sleeping apnea means you got a blocking nose, and this is why you snore when you're sleeping. And then, so this guy is always hypoxia because they don't have enough oxygen. They have lower sex drive and lower male hormone. And then we find that due to this according to this epidemiological evidence, sex ratio in human depends on this sex hormone level of father and mother during the conception. And then the higher testosterone level during conception would favor the subsequent birth of sons. That is to say, if you have apnea, that affect your sex hormone level. The second point is that if your sex hormone level is lower, and then they favor the birth of son, which is actually totally in line with the experimental evidence that we got from fish. Then we go further to ask the question, saying, will this pass to the next generation? Just like this cartoon. Say if the grandma and grandpa were subject to hypoxia, and then how about the son and the grandson? Just like this grandson said, grandma and grandpa, I'm innocent. I've never exposed to hypoxia. Don't pass your problem to me. Will that happen? So we asked another PhD student to do the work. Oops. Now, what we do is that we put the fish under normoxic condition, normal fish, grow them for 4 generations, F0 to F3. And then the other group we put under hypoxia. And then after 1 generation, we split this group. And then one group were also subject to hypoxia for the subsequent 3 generations. Whereas I mean, this is important. We take out some individuals which are subject to hypoxia for the F0 generation, put them back in normal condition, normoxic condition for another 3 generations. Then we try to compare their physiology, their sex, hormone, and so on and so forth. And we find out, in actual fact, that the hypoxia actually reduced the sperm mortality in F0 and F2 for the transgeneration group. That is to say, the sperm mortality of this, their son and the grandson decreased, despite the son and grandson have never been exposed to hypoxia, only because of the grandpa has been exposed to hypoxia. So this is what we call the transgeneration effect. Then we look at the percentage of mature sperm, spermatics, percentage of spermatic, and the same occur in the F2 generation, the transgenerational group. And then we look at the fertilization success. Again, the F2 are affected. I want to emphasize these guys and even the father have never been exposed to hypoxia before, and only the grandpa and grandma has been exposed to hypoxia. Then we then look at the epigenetic and transcriptomic and proteomic changes. We provide this evidence showing that in actual fact, they actually affect the fish, the F0 generation, affect the epigenome and change the epigenome, and then leading to this reproductive impairment in the F1, F2, and F3 generations. Now, then we ask the question, is that will the situation get better or worse? In fact, hypoxia is pretty simple. Everybody knows it, and it's because we produce a lot of these nutrients and put them in water. We produce lots of waste in wastewater, put them in water in the marine environment. It's extremely simple. We know about this problem about 30 or 50 years ago. But the problem in reality If you want to remove nutrient, it's very expensive. To give you an example, in the Baltic Sea, in the Scandinavian country, which is very rich, they reduce 50% in the N&P going to the Baltic Sea, which is hypoxic, and cost them something like almost 4,000 million US dollars every year. Whether it is affordable to some of the developed country, but definitely not affordable by the developing country. And then if you look at the growth globally, the growth of these treatment facilities are likely to catch up with the growth of population and industry, particularly in the developing country. And the other problem is that the contribution from atmospheric fallout non-point source are significant. Just like in the US, everybody knows about where the problem is, but because it's come from agricultural runoff, and then it's very difficult for the government to control agricultural runoff, because how do you know the nutrient come from me? And let alone, at least half of the nitrogen will go up to the atmosphere and come down. So they are not directly discharged in the river. So again, this causes major problem in controlling. And let alone, transboundary issue are difficult to resolve. If Germany is producing this nutrient and then affecting France, we expect sort of the Germany will sort of put in a lot of money to rectify the problem. The problem is not only with Germany, it's with the other country. Now, that's not the end of the story. Hypoxia, I'm sure you agree with me, is an evil, is it? And then when the meeting of two evils, when hypoxia meet with climate change, the situation will be even worse, because climate change increase in temperature would increase the freshwater runoff, increase in freshwater runoff, increase in water stratification, then enhance hypoxia. Freshwater runoff would increase nutrient flux, increase the productivity, and therefore also enhance hypoxia. The increase in the temperature would lower the oxygen solubility in water, and also increase the metabolism, again, add on the demand of oxygen and making the problem of hypoxia even worse. So overall, the risk assessment of hypoxia present to you is the environmental consequence is very serious. Area affected will be very large, economic loss very high, number of people affected very large, probability of occurrence very high, lasting effect, long lasting, takes months to years to recover, and the trend is getting worse. So we try to argue at the United Nations Forum and several years ago, and then we argue and saying that hypoxia is the most important problem, environmental problem faced, and so affecting our marine environment worldwide. And eventually we won. And then our work has been featured in the Science for Environment Policy by the EU, and also reported by all sorts of public media in US, Australia, and Nature, and worldwide. Then, to conclude, hypoxia is an old problem with new scientific insight and calling for an urgent reassessment. So for the first time, we discovered that hypoxia is an endocrine disruptor that can affect selected gene, specific gene, not every gene, receptor and neurotransmitter along all the HPG axis, and therefore causing reproductive impairment. Hypoxia is a teratogen leading to delay in development and malformation through disrupting apoptosis, affecting sex differentiation and sex determination, leading to a male-biased population and poses significant threat to the sustainability of natural population. Affects the epigenome, causing epigenetic changes of transgenerational reproductive impairment in subsequent generations. So given the fact that hypoxia occurs over a large area worldwide, and with this endocrine disrupting effect, teratogenic effect, transgenerational disrupting effect, hypoxia is the most important stressor in our marine environment, and it's difficult to control, likely to get worse, present a major threat to us worldwide in coming decades. And this is a research team from my previous research student, and working together with my research assistant, Professor Paul Lai, and then my colleague, Richard Gong. I present this talk in various occasions. And I said, if anybody can challenge me saying that hypoxia is not the most important stressor in the marine environment, I'll give them $500. I'm still keeping my $500. It's still valid. If anybody can challenge me and tell me with evidence saying that hypoxia is not the worst, well, you can get this $500 from me. Thank you.
Thank you, Professor, for the wonderful lecture. We have received a few questions from the chat box. Because of the time limit, I've selected 2 of them to ask. The first question is, do hypoxia exist on seagrass habitat? And what are the impact of hypoxia on marine plant? The second question is, can fish really swim away from hypoxia?
The first question is that whether they occur in the marine part?
In seagrass habitat.
In seagrass habitat, I must say, I don't know. And it's a good question because seagrass itself produces oxygen itself because it's produce a lot of oxygen that may counteract the effect. And nobody have done similar things before, I would say. But I would say that hypoxia, because it's so generic, they may affect the associated fauna and flora with these and the seagrass. But I don't know. For the second question,
can fish swim away from hypoxia?
Again, this is a good question. Well, if hypoxia occurs in a small area, the fish will swim away. But as I point out, hypoxia occurs in over very large areas. The fish cannot swim away. Like say, even in Hong Kong, if they occur in Total Harbor, the whole Total Harbor, there's no way the fish can escape. Let alone that occur in the dead zone, all these dead zone, as I said before, the dead zone in the Gulf of Mexico is the size of Virginia. If the fish want to swim away from the circle, hypoxic circle like Virginia, takes a long time, and then they'll die before they be able to do that thing.
Yeah, thank you, Professor Wu. The next speaker is Dr. Henry He, Assistant Professor in the School of Energy and Environment at the City University of Hong Kong. Dr. He is an aquatic ecotoxicologist who studies molecular and cellular toxicology using different aquatic invertebrate and fish models. Today, Dr. He will present a very new and interesting topic on liquid crystal monomers, which represent a class of emerging contaminants related to electronic waste. Dr. He, please.
Thanks, James. Thank you very much. Can you see my slide?
Yes, I can.
Yes.
Sure, no problem. Good afternoon, everyone. My name is Henry. So today I'm going to present a relatively new finding from our group called liquid crystal monomer, the LCMs, which is a class of emergent pollutants related to e-waste. So nowadays, I'm pretty sure everyone has a phone or phones, and together with a couple of electronic devices with LCD panels. The LCD panel is the major displaying and interactive media from human to those electronic devices. So you may know, since we have so many devices, so the global production of this LCD panel must be a giant number. You're right. So just in 2018, this is a bit old data, but in 2018, there are almost 200 million square meters of LCD panel has been built, synthesized globally, and the number is continuously growing, not surprisingly. So in 2019, in total, 53.6 million tons of e-waste was generated. So maybe roughly at least 1/4 of them contains some sort of form of LCD panels. And particularly after 2019, after the outbreak of COVID-19, the demands of study, work, and even entertainment at home has already greatly boosted the sales of consumer e-devices. You may heard about currently the supply chain problem in the United States. And part of it is due to the shortage of all these electronic devices. If you recently order a laptop, you may suffer from a delayed shipment. That's because the greatly boosted sales, the total sales. So you may from this information you may predict that the production of the LCD panel will continuously increase even during the COVID-19 pandemic. So now let's move to the substrate, the LCM. So LCD panels can actually emit many organic compounds during usage and disposal. Some of them are VOCs, and these will include the LCM, liquid crystal monomers, which is the essential substances used in LCD panel as the displaying material. So up to now, according to our survey, there are more than 360 LCM have been synthesized for commercial application. More than half of them are considered persistent and bioaccumulative. And about 1/4 of them are very persistent and very bioaccumulative based on their physiochemical properties. So actually, for one single model of panel, there are usually more than 10 to 20 different kind of LCM to form a mixture. So the manufacturer will apply this mixture to form the cocktail of liquid crystal and then fill in to the panel to form this LCD panel. And this is the typical LCM used in a lot of model of laptops. So a typical LCM consists of a hydrocarbon backbone with several aromatic rings. The length of the molecule is generally larger than its diameter, and the multiple bonds along the long axis or a system of conjugated double bonds will make the molecule rigid and planar. So it's a planar molecules, and modification of functional groups to are normally used to enhance the performance of the molecule, such as introducing a side change, replacing phenyl groups with cyclohexyl groups, substituting phenyl ring hydrogen with other functional groups, particularly the fluorine. Yeah, you can see the fluorine here. So according to our study, there are and some other group study, there are a couple of different classification of LCMs. You can see it's here. So the first one is by cyclohexyls and its analogs. You can see there are two cyclohexyl ring here. The second group will be biphenyls. So we will have two phenyl rings here. Cyanobiphenyls and analogs, and the last group is by cyclohexyl phenyls analogs. So these are the four major groups utilized in the common LCD panels. So these functional groups will contain, you can see the R here. So these functional group will usually contain cyclohexyl ring, additional cyclohexyl ring, or phenyl ring, or diphenyl acetylene, phenyl nitro group, or fluorophenyl group. So all these terminal functional groups makes the molecules persistent, bioaccumulative, and biologically toxic. So we call these substances PBT substances. So the concentration of LCMs were comparable, according to the current study, very limited. They are comparable to those of legacy and emergent organic contaminants related to electronic devices or e-waste. They're including PCB, TBDEs, OPFR, that's organophosphate flame retardant, and NBFR, the novel brominated flame retardants. So they are all at comparable level, although the application of LCMs are relatively new. So a recent study studied 17 LCMs, which has been quantified in the indoor surface dust. So they collect the surface dust like on the table, on a wall. So the total LCMs range from 0.13 nanogram per gram to 2,210 nanogram per gram, with a very clear correlation with the usage extent of e-devices, which means if a room has more LCD panel or other related devices, they will have higher contamination level of LCMs. In another study, in total 14 LCMs were determined in sediment samples collected near one e-waste recycling facility in the Yangtze River Delta region, with total concentration up to 26.3 nanogram per gram. This is not a very high concentration, but mainly because that e-waste facilities is very young. It's very young. In the Sweden study, this group detected a wide range of indoor organic contaminants. 6 of them are LCMs, actually. So the predominant species, I didn't show up the structure here, the MEO3BCH, with the concentration ranging from a very high, actually 68.6 to over 1,500 nanogram per gram. So this particular species actually are also determined in this first study, the sigma 17 LCMs, but with very low concentration. So you can see the dust in China and dust in Sweden are different regarding the LCM profile. So it's a very interesting study. And very recently, another group determined 34 fluorinated LCMs in a dust sample collected from e-waste dismantling center, which is in Guangzhou. So as you may know, the Pearl River Delta is the center of e-waste recycling in China, maybe even all over the world. So that's the very high density of LCMs contamination in this area. So in that study, the total LCM concentration will be up to, oh my God, 976,000 nanogram per gram. So it's pretty high. That's in the recycling facility. So recently, the same group actually determined 39 fluorinated biphenyls, 19 biphenyls, and 6 cyanobiphenyl LCMs in waste computer television LCD panel. You may know when you break up LCD panel, you will see a lot of dust in the panel, right? So actually, those dusts are seriously polluted by a variety of chemicals, including LCMs. So the total LCM concentration range from 50 something to 2,000 microgram per cm square, and more than 36 of them were fluorinated LCMs. So the toxicity, let's move to the toxicity. So the current knowledge of the toxicity of LCM is very minimal, almost minimal, nothing. So liquid crystal manufacturers, so those manufacturers claims that LCM are non-irritating to skin and eyes based on mammalian models, so the rat and mouse. However, a very early study, it's published on liquid crystal, so not in any environmental science study. So compared to the leotropic promonic LCM, the thermotropic LCMs, and surfactant-based leotropic LCM, so the 3 different kinds of liquid crystal. So the later 2 show significant cytotoxicity based on the bacterial viability acids. And another early study, so this Korean group used a wash-off mixture from broken laptops. This mixture contains a high level of LCM. By that time, they didn't know what kind of chemicals cause the toxic effect, but this mixture actually caused significant adverse effect in fish, including impaired antioxidant enzyme activity. So in the first systematic study of LCMs, Professor Seuss discovered exposure to LCM extracts from cell phone screens significantly impair a battery of key genes. He used the chicken embryo. So particularly the upregulation of CYP1A4 up to about 14-fold indicated dioxin-like toxicity of LCMs. So LCMs, particularly the fluorinated LCM, are potential persistent, bomb cumulative, and toxic contaminant, PBT contaminant, However, similar, you may recall the story of PFC, like PFOA and PFOS, that's in early 2000s. The current extent and magnitude of the environmental distribution, toxicity, and health impact of LCM are largely unknown. That's the very similar case compared to PFC in early 2000s. A lack of standards analytical method, right now there's only a couple of studies try to quantify this kind of chemicals. So there's lack of standardized method and data for sure to detect their environmental level, especially in the aquatic environments. You can see I present several literatures about dust, but there's no, particularly no marine data. Currently, we are doing one, and hopefully we can submit the manuscript very soon. And some LCMs analog do not even have reference standards. There's no standards for it. So we have to use suspect and even non-target analysis approach to determine these chemicals. The source of LCMs in the environment is not well understood. So it might be from industrial effluent or the domestic emission. As I said, during the normal usage, these screens can also emit LCMs and may be contributed to the environmental pollutions. So LCM, particularly the fluorinated LCMs, may display similar toxicity as PFC compounds. However, their ecotoxicity and human health risk data are absent. So given the potential enormity of the contamination issue, like the e-waste issue, everyone knows the entire Earth is surrounded by those electronic devices from domestic usage and laboratory usage and entertainment usage, you name it. So filling the above knowledge gap is urgently needed for a comprehensive risk assessment of LCMs as an emerging group of contaminants. Now let's move to case study that we are currently, we just published it. So we put our focus first on the landfill leaching. So you may know, due to inadequacy of proper management, particularly in the early time, massive amounts of e-waste has actually been discarded into normal municipal solid waste landfill. They're like ordinary municipal landfills. So many studies, actually a lot of studies, have reviewed that e-waste related pollutants, you name it, like PFOS or PBD, all this stuff can be detected, in the landfill leaching and the soil, groundwater, surface around the landfill sites. So there is clearly a migration of these contaminants from leachate to the environment. Therefore, landfill leachate is not only a potential sink of the e-waste pollutant, but may also act as a potential point source to promote these migration of these pollutants into the local environment. So it's since everywhere has landfill sites. So as far as I know, Hong Kong currently has 3 active landfill sites, and more than a dozen of landfill sites has already been decommissioned. So currently we are working on a large survey on the including a lot of landfill sites in Hong Kong, China, mainland China, Korea, and other places to see the distribution of these chemicals. But this is definitely not a local problem. It could be global. So the aim of this project is to develop a robust analytical method to determine 38 target LCMs in leachate sample, which is actually very difficult. It's a very difficult job to do. And second is to determine the concentration and distribution of LCMs in leachate sample collected from 2 typical landfill sites in South China, which is one in Shenzhen and one in Hong Kong. And third is to raise the concern of these issues, like the landfill is at the potential point source of this emergent e-waste pollutant, and urge further research on their occurrence, migration, toxicity, environmental risk, particularly in the aquatic environment. Because if we don't know whether these chemicals exist in the landfill leachates, we will not put specific effort to remediate it, right? So it will be likely discharged directly into the environment, and particularly the marine environment, to cause problems. So we have 38 LCM chemical, 1 internal standards. Most of them can be purchased, some of them can be purchased from Tokyo Chemicals, but a lot of them we have to directly contact the liquid crystal manufacturers. So there are 2 leachate samples from Hong Kong and Shenzhen. So you may know Shenzhen is just alongside Hong Kong. 2 cities are very close to each other. So a combination of ultrasonic enhanced liquid extraction coupled with saponification and silica gel packed column purification was developed for the pretreatment of the samples. And we use GCMS to analyze the samples. So here are some results. I want to keep it as simple as possible. So the first one, the A, is the recovery. You can see we did 2 nanogram, 10 nanogram, and 50 nanogram spiking. So we spike non-level of these compound into leachate, and then we will measure the real concentration. So at the end, we chose 10 nanogram for the spiking. So we can generate a standard curve to measure, to determine the real concentration. B shows the UV254, which is equivalent to like total organic compound measurement. So you can see after the ULLE means ultrasonic liquid extraction. So you can see it significantly decreased the overall background of interference of other organic compounds and keep our target compound, the LCM, in the extras. So this figure showed the different performance of cleanup procedure. There are saponification, acid wash, silica gel column, whatever. It is also shown that all these clearance procedure can significantly remove a large proportion of we call matrix effect, ME. So combine them together, we develop this ultrasonic enhanced liquid extraction coupled with saponification and silica gel purification. So we, after all, we receive around 80% to 100% recovery for all the target compounds. So here are the results. I don't want to go to detail, but there are some key points. So in Hong Kong leachate sample, the total LCM concentration are more than double of the Shenzhen compound. You can see it's 1,120 nanogram per liter. Shenzhen is about 4. Oh, let me use this pointers. Yeah, Shenzhen is about 409 nanogram per liter. And among them, there are more fluorinated LCMs in Hong Kong's samples, more than 600, and in Shenzhen is less than 50%. So there's a significantly different profile between Hong Kong sample and Shenzhen sample. So 2 cities are different. So more interestingly, for example, for this A figure, we detected 9 fluorinated LCM concurrently in Hong Kong sample and Shenzhen samples. So these are the 9 LCM, fluorinated LCM, and they show a very nice correlation, which means whatever fluorinated LCM detected higher in Shenzhen sample, it's also higher in Hong Kong samples. Okay, so that strongly indicated that the mixture of these 9 fluorinated LCMs are widely used in certain popular model of devices. I'm not saying it's from some phone, but it's likely from those very popular brands of devices that result in the same combination of these compounds in the landfill leaching. And the right figure just show the concentration range that we detected in our current study. So which is almost comparable. It's comparable to other study targeting the landfill leachate. You can see this is the current study for total LCM. These are PBDE, so the LCM is already higher than the total PBDE. So the problem is real, I have to say. So the magnitude and impact of LCM pollution is expected to be significant. So there are a couple of future work that we are trying to do, and we also want to promote this topic to many other groups so we can work together. So the first thing is that we want to expand the LCM standard bank for target analysis. Currently, we have secured a bank of 64 different species, and I believe the number can be increased further. And second is to develop methods for suspect and non-target analysis. So there's, as I said, there are more than 300 different chemicals for LCMs, and even more without standards. So we need a non-target approach for sure. Determine LCMs and analytics in various environmental matrix and even biota samples, like fish, for example, even marine mammals. Currently, we are working on, one of our students working on dolphin. So the LCMs in dolphin levels. So even for some basic toxicological data, it's missing, like LC50, the basic LC or EC50 for most of the LCM, there's nothing. And the computational toxicology can also help for prediction of those basic toxicity data. And moreover, the sublethal toxicity and toxic mechanisms are not very well understood, including biotransformation, genotoxicity, lipid metabolism, neurotoxicity, reproductive toxicity. The list could be a very, very long list. So definitely there are a lot of works can be done in the near future. So at the end of my presentation, I would like to thank my team and the support from School of Energy and Environment and the Stakey Laboratory of Marine Pollution. So this work has already been published on JHM. If you have any further questions, you are welcome to email me. Thank you, everyone. Thanks, James.
Yeah, thank you, Dr. He. We are slightly behind the schedule. We have one very quick question by Professor Wu.
Yeah.
Hi, good talk, Henry. And could you tell me what's the approximate KOW value of all this compound?
It's comparable to PBDE. Yeah. The range is very similar. Yeah.
Okay.
Okay,
thank you. I presume that you're also working on the toxic equivalence and things like that.
Yeah, we are currently working on it.
Okay, thank you.
Thank you. So James, I will give you the podium back.
Yeah, sure, please.
Sure.
The next speaker in this session is Dr. Venkateshan Tyagarajan. Sorry for my bad pronunciation, Dr. Rajan for short. Dr. Rajan is an associate professor in the School of Biological Science at the University of Hong Kong. Dr. Rajan has developed a broad research expertise, for example, in environmental proteomics, biomineralization, biofouling, and oyster aquaculture. Today, Dr. Rajan will share with us his important findings about the Chinese oyster and their responses to ocean acidification. Dr. Rajan, please. Hello, is Dr. Rajan here? Hello. Dr. Rajan may be upset about my pronunciation of his name. Is Dr. Rajan here, please?
If you won't mind, should we move the thoughts forward or we take a longer break? Which way better?
If you ask me, I would suggest perhaps we have the next speaker presenting first to see if Dr. Rajan will come back on time.
That is me. So
Yes, I know.
That's good. That's good.
Okay. The next section will be chaired by Professor Kenneth Leung. Professor Kenneth Leung.
Huh?
Or do you have the break first?
No, I can give the talk now.
Okay. Yes, please. Professor Kenneth Leung.
So I'm going to talk about eco-engineering for enhancing marine biodiversity. So let me share my slide with you. Can you see my slide?
Yes, I can.
Good. So in the coming 20 minutes, I'm going to tell you about the principle of this eco-engineering, how this can help to increase marine biodiversity on artificial seawalls. So as you know that seawall is very important to protect us from the wave action and also flooding and soil erosion. They play a very important role in urban structure. So however, if you look at the typical seawall here, we have many vertical seawall is for the boat parking. And also if the area is mainly for open area for irrigation without boat parking, then you can use this as sloping whip-web seawall. If you take a closer look on those seawall, you cannot find too many marine life. Most barnacles, oysters. Sometimes you may see some leakier, those are isopods. Then you may ask why the biodiversity is low in those artificial seawall area. So if you look at this thermal imaging, the upper one is the vertical seawall. When there was a low tide in the midday with strong sun, then the temperature can go about 50 degrees Celsius. That will kill many organisms. On the contrary, if you go to the very nearby lateral rocky shore, then you can see the rocky shore with lateral erosion. It has some indentation, crevices, and up and down structure. Then turn out the temperature in these features could be 10 to 15 degrees Celsius lower than the top, those highly exposed area. So take example from this lateral shore, then you can see all these white color patch are the dead barnacles scarred on the rock. This due to the heat stress and also the desiccation stress. If they live here at the low time in the midday in summer, they will be killed by thermal stress. But you can see many mussels here and also barnacle here. They go really well because this area can trap the water, allowing them to have a much better environment, cooler area for them to survive. As you can see, many animals actually like the crevices area as well. So another aspect is important to talk about. When you go out to the seaside with your girlfriends or boyfriend, you walk on this rock surface, then you can see the color. Sometimes it's brown color, sometimes it's green color, some are red color. If you scratch on the surface and then using a microscope, you will find them, they are plant species, they are microscopic plant, like a diatom and also the cyanobacteria. They are very important food for the grazer, those who eat vegetable, like many of us. as you can see, this chitin and limpet and this periwinkle, they all fit on this biofilm. You can see after they graze on them, the biofilm gone. So this is a very essential element for the rocky shore. If the temperature too high, this biofilm will die also. So take a look at this diagram. First of all, the seawall, you want a seawall to harbor more species first. The seawall have to be warped so that it can anchor the nutrients, protein, carbohydrate, and so forth, and then attract the bacteria to go to the biofilm, and then subsequently the plankton, zooplankton will come into place, and then finally the whole community will be formed and attract fish and crabs to use this habitat. So we try to develop different types of eco-engineering design product. As you can see here, if you have just a very, very smooth surface and very warped surface, then you can see even the same tidal levels, same environment, not much life on this smooth surface. But you can see many life, they like this warped surface. In actual fact, this is not new because when Professor Wu was young, when he did his master's degree at the University of Hong Kong with Professor Biomotor, he worked on barnacles. He told me that he can make a heart shape so that the barnacle will follow the heart shape to attach. If you make the concrete surface rough, they will just attach to it. The same idea we use here, we make groove and up and down structure, then the barnacles really love it. Not just barnacle, but also the algae and also the oyster, they love this rough structure because it can retain the organic matter and then allowing the formation of biofilm. And then the animals love to live on this kind of nutritional rich area. And we also find another problem is the use of concrete. Most of these cement for building purpose, they have high pH, around 13 or 12. Why? Because they need to protect the reinforcement rebar, the metal bar inside, so they have to retain the pH as high as possible to reduce corrosion. And also when they do this manufacturing process, the surface in most cases are very smooth. It's not very good for the marine organism to settle on. So to rectify this problem, then we have to modify the surface to be very rough. And also we have to reduce the pH to closer to the marine environments. The seawater pH is normally 8. So we have to reduce the pH to that level, if possible, using a carbonization technique or using different concrete mix. At the end, we cannot use metal rebar, so we have to use a glass fiber as the reinforcement structure. After that, then we can build the Lego, say, with the shading area to cut down the temperature, with crevices and water retention elements so that the animal can have a more benign or better environment to live. One example is quite interesting. In Hong Kong, in Sai Kung, this is called Yin Jiam, the place. There was one accident, a barge sank at the bottom due to a traffic accident. It carried a lot of bags of cement. And then after more than 35 years, you can see that not much life on that because it is smooth and also high pH, no those microhabitat for animal to use. So then if we want to make the concrete to be livable, we have to mix the above mentioned changes. So after we design this kind of new eco-engineer system, then you can see if we have a whole, like a tube structure, the penny fish, they love it. Then you can see the crevices we create, the cowrie and the periwinkles, they love it. They also the dog well lay the eggs inside those burrow area. Then the whole structure after half year to 1 year, you can see a very rich with mussels and spongy, and also sea anemone. The crabs really like those crevices. This technology actually have been booming over the past decade all over the world, from UK to Seattle in US, Sydney in Australia, Florida in USA, and also in Israel. They realized this problem of the desert in seawall. Now we can improve the situation to enhance not just biodiversity, but also fishery resources in urbanized coastal area. So if we do it properly, we can allow 3 major services for the seawall. First, shoreline protection. Second one, we can enhance the ecosystem. Finally, if the area big enough, we can allow people to enjoy the waterfront, okay? So if we do well, then we can enhance biodiversity, reduce biological invasion. Some studies showing that with the engineered ecosystem, then there will be reduction of invasive species because there will be more predators to control them. And then if we build the eco-engineered shore here in between 2 lateral shore, then we can increase the connectivities and allowing better gene flows in the entire ecosystem. So the settled down organism like mussels, bivalve, oyster, tube-worms, they can also filter the water to clean up the water, eventually improve water quality. So if we make the eco shoreline longer enough, it can help to improve fishery resources and also fix carbon as well. So if the area big enough, like this one in Sydney, they use this lateral sandy shore, this sandstone. You can see after millions years of erosion, then you can see many holes and structure there, allowing the marine organism to settle. Those are very lateral type of rock. Then people love this park because people can sit there, enjoy the waterfront. During the low tide, the kids can go down to see the marine organism. Now the Hong Kong government just copied this design into Hong Kong now. In North Pond and also in Causeway Bay, we are building similar type of steps in Hong Kong. So now come back to the science. We all know that if we increase the capacity of the ecosystem, we're going to have more different species to live in. Just for example, if you have a farm field, just those grassland, then you have only few species. If you start planting different trees with different fruit and different leaf structure, and then you will harbor more different mammals, birds, and insects. And then you keep doing this and then make it like a mosaic, then you can enhance the biodiversity, overall biodiversity greatly. So then in this study, we try to test this hypothesis. Then we have a very simple structure. This is a control, nothing, just a concrete tile. And then the next one, we have this ridges coming up 2.5 cm. And then the next one is more compact, 5 cm. Is a much deeper ridges. And then we clean the seawall as the reference site or control site. And then side by side, we also want to test this hypothesis called, it's called lateral assemblage. So we just stick the rock oyster on top of these 3 different designs to enhance the capacity. So the capacity increased from here to here and also from here to here. Then we try to compare to 2 different sites as well. Then we install them on the vertical seawall and compare also with a reference site in the lateral rocky shore next to the seawall. The entire study lasts for 1 year. Then we have monthly data and also the final study. We took out the tower and then do a very detailed study on everything. So it was a bit of surprise that we find many species we never seen on the controlled seawall before, like these crabs, and also these amphipods, and also these bivalve. So it means the new habitat working really well to harbor new species. Then it answers our question that the new structure can increase biodiversity. So after 1 year, when we look at the results, we can see from the fact 1, 2.5 to 5 cm ridges, we can see increase of number of species. If we put the rock oyster on top, then the increase even higher. Same as the other side, Lo'ompai, same trend, okay? However, sorry, for the abundance, for the same site, Lo'ompai. But some 水果 we only see in the one without the oyster. The one with oyster, we see the increase, but without those a pattern linked up to the tower capacity. So we find that in some 水果, because it's a low west facing, during the low tide summertime, it's facing to the west. It's always have strong sunlight. Then the capacity couldn't really cool down well. So that means we won't see this higher increase when we compare to this site. So the orientation of the device also need to take into account. So we will learn from this. Now we design something that will use the back side of the tower rather than in front. The front side is like an umbrella to cut off the light and heat. Now we try to use this new system. So if you look at those with the seeding of the oyster, the rock oyster, you can see they can harbor more oyster. The babies will join them at the end. So this is a cheap and good method. If you can't afford to buy this engineered product, you can actually stick those oyster on the seawall. Then guarantee you will have more oyster babies come by and also more other species to join the ecosystem. And we also find most species, they like to see in the bottom of the crevices rather than on top of the ridges. They try to hide away from predator. At the same time, it's a much cooler places with more food in the crevices. So overall, we find that with this just the capacity made by the tower itself, then the number of species can increase by around 20 to 50%. And with seeding, then the increase would be around 40 to 75%. Then for the abundance, the increase even more massive, can increase 4 to 5 times, depending whether we add the oysters. So then this experiment demonstrates that this simple technology can enhance the biodiversity quickly. Then the European Commission also adopted our result as part of their Science for Environmental Policy to inform their leaders to also try to adopt this new technology in urban infrastructure. So our project actually joining with the World Harbor Project. In these 14 cities, we're doing the same experiment together. Then the result almost very identical, except that we found in tropical area, the recovery of mobile species is higher, but in the temperate region, then the recovery with the engineering tower, we see more sessile species like barnacles and seaweed. So some differences, but we still can see the enhancement of biodiversity due to the ecosystem capacity constructed by the multi-flight towers. So now I would like to share more little bit complicated concept. So in this diagram showing that x-axis, when we have more development disturbance, then the beta diversity means number of new species will be declined. So then we try to reverse this using eco-engineering by pushing this curve backward. Imagine if we create more different types of habitats, then we should see this reverse curve. We can see enhancement of biodiversity. So we try to test this hypothesis using more different devices this time. We implement really big panel, 1 meter by 2 meter. And this eco tower more complex decided by our team and Polytechnic University. And the oyster shell basket, plus the tidal pool, and also this armoring unit. They presenting different type of new habitats we create on the artificial shoreline. So you can see all the structure is really big, 1 meter by 1 meter by 1 meter for the tidal pool and also the armoring unit. These are the basket, this 1 by 1 meter with ½ meter depth, okay? Then we installed them for 2 years. The project just finished. Then I just show you some of the results. This is the oyster shell basket. We put bags of oyster shell into the cage, forming a more natural habitat. Then you can see many birds like our artificial seawall here on our structure, because sometimes they can have easy meal to take the fish from our structure. This is the overview aerial photo of our devices. Then we do the field measurement every season, once every 4 months. We are doing the survey. Then this just a 9 months data, but you already can see the trend. The pool and armoring unit better than the control seawall, and the panels also perform better in terms of species richness. Now the results are even more pronounced after 24 months. But we do find the winner actually is not those concrete structure. The winner is this oyster basket. We just put live oyster or just empty shell, the dead oyster shell. They can harbor more than 70 species in one site. The result just amazing. And then because the cage itself as a protection, protect the animals from predation, then we can find many different species inside the cage just after 1 year. So as you can see, just 9 months, you can find on the live oyster, we found 20-some species, same number of species found on just shell. So what we suggest here, we don't need to put live oyster, we just need to put the shell. You will find a similar species harbor on the shell. Then the cage is important as a protective environment. Then you can see another 20+ species joining. So come back to my earlier mentioning this reversal of this curve. Then we just use one site as example. Each column represent, this is just the ordinary seawall. And then we add one device, the pool and then armoring unit. Then we find new species, we add on the curve. Then you can see the curve actually pushing backward this way. That means if we create more and more different habitat, we can harbor more new species to enrich the overall biodiversity. We are doing more complex analysis at the moment with the 24 months data from 3 different locations. So hopefully, maybe in a few months' time, we can share the final result with all of you. So the key driver why we are interested in this project, because the mainland China, Chinese government, our nation, want to do big scale ecosystem restoration across the whole country, from rainforest to deserts to the marine environment. There will be many major projects for protection and restoration of coastal ecosystem, especially in our region, Greater Bay Area. The government encouraged us to implement this eco shoreline or eco engineering of shoreline implementation. Then Hong Kong government have been implementing this 3.8 kilometer long eco shoreline. I was invited to be one of the designer of this new eco shoreline in Dongchong next to our airport. So in the area closest to the river mouth, the estuary, we have the mango plantation ecosystem. And then in the exposed shore area, we use this eco-concrete to build different sizes of pools and with different structure I showed you before. We already implementing a trial. The trial result really good as what we predicted. So then we also won important design prize for the sustainability award. So we actually we start from nothing. I was new to this business. When the government thought about eco shoreline engineering, they have no idea. So they asked me whether I can help. So I told them that I need some help from outside. Then I invite all the leaders in the field. In 2016, we organized the first workshop to learn about the concept and the challenge. And also they give us advice on the design of the Dongchong eco shoreline. And then in 2018, I even invited more people, 28 people, 25 experts to join us. Then I locked them up for 4 days. Then we wrote these very, very important papers together. And this paper summarized all the theories and practices and consideration and also assessment criteria all in one. Then if you want to know more about these topics, you can use this QR code to get a paper from us. Then in coming January, we will have another workshop. Then we will focus on the develop the specification when we develop this kind of eco-engineered product. So then you all are welcome to join us. The conference will be in hybrid mode. Then from overseas people, you can join through the online system. And also, since the conference is also part of the United Nations Decade of Ocean Science Action, so then we offer some free registration places. Please contact our conference secretary, and we welcome you to join us. So finally, I would like to thank all my partners and also most important, our sponsor, CEDD. Thank you very much.
Yeah, thank you, Professor Leung, for his wonderful talk. Is there any question from the floor, please? If not, let me ask a quick question. Eco shoreline is a very good concept to restore marine ecosystem on man-made seawalls. How do you compare the effectiveness of this artificial structure with other more natural restoration means, for example, planting mangrove, a strategy used by some audience here in the Philippines?
So if you try to restore a natural shoreline, that is easy to do the mango plantation. It will work well. However, we're talking about this kind of reclamation is different ball game. In a natural shore, you have the inclination from land to water gradually. But when we talk about reclamation, you like this. It's drop that. In fact, what the seawall you can see is just like the hanging out like this, and then it go all the way straight down 20 meters. So if you want to plant mangrove, that means you have to sacrifice some land footprint so that you have a extension like a gradual slope. We did it. We are going to do it in Dongchong, but still we're talking about 3 to 5 meter stretch area. Is not too big. If you want to have a very nice mangrove system with outwelling and succession, this is not feasible because you only have a limited space. So then in that case, we have to use a combination with those concrete-based engineering techniques joined together. But as I show you in our result, we find that the oyster shell inside a cage is the best. When we compare to engineering product, they may harbor up to 30, 40 species, but the oyster shell cage can harbor up to over 70 species. So that means in the way forward, we will try to use the oyster shell more, put them inside some lateral fiber rather than using the metal cage. Hopefully they look like more lateral in a sense. So we just got a grant to deploy oyster shell bags in Lemma Island. So we will try this out.
I see. Thank you, Professor Leung. Wonderful talk. This is an announcement. We have a slight change in the schedule. We will have a break now. We have a break now, let's say 5 minutes. 5 minutes. After the break, Dr. Apoptosis will give her talk on coral restoration, followed by Dr. Rajan on oyster aquaculture under ocean acidification.
Okay, thank you, very good.
Okay, 5 minutes break. Thank you. See you in